Good afternoon, everyone. I'm Sureni from Julia Dahlman's lab. Today, I will be presenting my work on sensory-induced hyperactivity associated with SYNGAP1 AB zebrafish models. A human brain has billions of neurons and many of them fire at a given time. In order to maintain normal brain activity, it is important to have excitatory and inhibitory balance. If excitatory inputs exceed inhibition, one might experience epilepsy-like phenotypes. If the inhibition is increased compared to excitation, those individuals might experience sensory deprivation. This is a schematic representation of an excitatory synapse. So many of these proteins that function at this postsynaptic density are known to be associated with ASD and ID. One such protein is SYNGAP. So when the neuron is at rest, SYNGAP inhibits these small GDPases, such as RAS, but when the neuron is depolarized, there will be an influx of calcium ions, which would activate CAMP kinase 2. CAMP kinase 2 will in turn phosphorylate SYNGAP1, so that is the activated version of SYNGAP. So activated SYNGAP can no longer inhibit these more GDPases. As a result, we get increased ERT signaling, which in turn lead to increased translation. And also we get AMPA receptor traffic into the membrane. So this part is very important for synaptic modulation associated with learning and memory. During the last decade, a growing number of loss of function mutations in SYNGAP1 have been discovered to cause ASD and ID, and these mutations span throughout the protein. With that, we ask the question, how do SYNGAP1 mutations impact nervous system development and behavior? In our lab, we use zebrafish to study, to answer these questions. So why zebrafish? Zebrafish have sequence similarity and conserve genes with humans. They also have excitatory fertilization and optical clarity, allowing us to study early developmental processes. We can also use comparatively inexpensive gene editing techniques such as CRISPR-Cas9. Given their high fecundity and small body size, and also highly conserved stereotypic behaviors, make them a good model to carry out these high throughput behavioral assays. With the available tools, now we can do whole brain activity mapping to identify brain regions associated with different behaviors. So throughout this talk, I will be referring to SYNGAP1A and SYNGAP1B. That is because zebrafish have two SYNGAP1 genes. That is because teleos went an additional genome duplication event, thereby retaining two copies of SYNGAP1 gene. When we look at the proteins in both SYNGAP1A and B, they have the same domain structure as we can see in mammals. So we use CRISPR-Cas9 genome editing to create these loss of function mutants, and these red bars represent where we find the mutations. A lot of work has been done in the past few years trying to identify different mammalian isophones and their respective functions. So this work inspired me to look at what are the zebrafish isophones and annotate them. So the way that I did was I obtained zebrafish protein sequences from NCBI. So those included both published and predicted sequences. And then in order to identify unique and common exons, I used UCSC zebrafish genome browser to test whether they are expressed in zebrafish. I further tested them against using expressed sequence libraries such as ESTs and TSAs. After that, I was able to identify 5 different SYNGAP1A isophones and 7 different SYNGAP1B isophones. Similar to mammalian isophones, both these genes show variations in their C and N termini. and their core exonic regions remain somewhat intact. Interestingly, I was also able to identify a SYNGAP1B isoform, which is this orange one, which has over 95% conservation with well-studied mammalian alpha-1 isoform. So this isoform also had a putative PDZ domain, suggesting that this might be the isoform that is enriched at the excitatory synapses of the brain. So just like with the mammalian models, after coming up with this figure, it made me wonder how differentially expressed these isoforms are, where are they expressed, and what are the functions that they serve, because we have two different genes here. Okay, so trying to understand what are the phenotypes associated with the models that I have. So since human SYNGAP1 variants experience sensory-induced hyperactivity, I wanted to see, using zebrafish, whether they also exhibit similar kind of phenotype. So in order to do that, we placed SYNGAP1AB heterozygous larvae and wild-type larvae in a 96 well plate, and then we placed them in a behavioral chamber with a camera to capture larval behavior. So first, we gave them, we dark adapted them before exposing them to 5 minutes of lights on and 5 minutes of lights off. So this is what the output would look like. So to your left-hand side, you will see wild-type larval activity, and to your right-hand side, you will see heterozygous SYNGAP1AB larval activity. So this is a speed video at 16x. And also you will be able to see two different distinct phases where there is less activity and then there is increased activity. So yeah. Sorry about that, that freezes for a bit. See, this is low activity levels and this is high activity levels. Okay, so then we quantified this activity that we see. So in these graphs, always blue that is coming from wild type, red is data coming from SYNGAP1AB heterozygous larvae. So y-axis, it's always time, and then we graphed mean distance moved by a larvae during light on periods and then during light off periods. So one feature that I would like to point out that, so these three are coming from three independent trials, but you can see during dark phases in all three trials, the activity levels are much higher in heterozygous larvae. However, during lights on period, it varies a lot, the activity varies a lot. We think this might be due to a batch effect. Given these observations, we are currently working on identifying possible levels at which SYNGAP1 function in sensory motor circuits in zebrafish. In order to help address that, we are working on whole brain activity mapping to identify the regions that are associated with these visual stimuli and whether there will be any differences in brain regions or activity levels in SYNGAP1 larvae compared to wild type. Okay, given that ERK pathway is downstream of SYNGAP1, I would expect to see increased phosphorylated ERK in heterozygous mutant compared to wild type larvae. So, so this is what it's going to look like. So we will stand for p-ERK activity after exposing them to a stimulus, and then we also would stand for total ERK, and then we will normalize p-ERK with 2 ERK, and to get the significantly regions that are significantly different when like in the presence of a stimulus. To further test loss of functions in GAP1 models in terms of their learning and memory, this is another experiment that I'm working on. So this is a habituation assay based on Owen Randler's work, which uses a dark flash as a stimulus. So here, this graph shows how habituation occur in wild-type larvae. So from this assay, I would expect SYNGAP1 heterozygous larvae to show reduced habituation or no habituation due to their altered synaptic modulation circuits. So to summarize, SYNGAP1AB zebrafish models of AHD-ID will help understand roles of SYNGAP1 in development of the neural circuits, which in turn will help us address the mechanism underlying ASD-ID linked behavior. So with that, I would like to thank Dolman Lab members and zebrafish facility manager, Ricardo, for his support. And I would like to thank the funding agencies, NIH, NICHD, and University of Miami Department of Biology for all their funds. Thank you for your attention, and if you have any questions, I'll take them. Thank you.